Hi team, I'd like to talk a little bit about genetic engineering in animals today. In our other videos on plants, you'll have talked about genetic engineering versus GMOs. In this video, I'd like us to talk about the use of homologous recombination to introduce new genes into animals. And I think we'll also compare and contrast transformation of fertilized eggs or embryonic stem cells that have not yet been fertilized. Ultimately, with this information, I hope that you'll be able to argue for or against the use of genetic engineering. First, let's talk about some genetically engineered animals. The first one is the spider goat, or rather, goats that have been engineered to carry a gene that produces a protein found in spider silk. And this spider silk protein is one of the strongest, most hardy, sturdy substances known. It's hard to get enough silk from spiders, even on a spider farm, but by producing this protein in goat's milk, we can harvest quite a lot of this spider silk protein and use it, for example, in the production of protective gear or almost armor for our soldiers. Importantly here, guys, we're not milking out strings of spider silk from these goats. Instead, the protein is found within the liquid milk of the goats. Another really interesting type of genetically engineered animal is the Aqua Advantage salmon, which has been approved for human consumption by the FDA, and we'll be reading a paper about this in just a bit. This image shows two fish. On the bottom here, a normal 1-year-old Atlantic salmon, and on the top, an Aqua Advantage salmon, also 1-year-old, also an Atlantic salmon. What has been changed here is the addition of one gene, a growth factor gene. You can see that the Aqua Advantage salmon grows twice as quickly as the regular Atlantic salmon. And these fish are only farmed, they're not grown in the wild, and they're actually all female and sterile. Have you heard of that before? That's what they did with the dinosaurs in Jurassic Park. Now, Aqua Advantage salmon probably won't wreak the same type of consequences as a free Tyrannosaurus Rex, but they have been approved for human consumption, and the hope is that these farmed salmon would allow us to get more meat more quickly and have less of an impact on the natural populations of salmon. Another type of genetically engineered animal is the mosquito that is carrying a version of a gene that prevents transmission of viruses such as malaria or Zika or dengue fever. And these mosquitoes often carry a gene drive that will help push that gene through the population of mosquitoes. This is something that does occur naturally, but we can talk more about gene drives when we get to the CRISPR portion of the course, as gene drives are really a form of genome engineering and not just of transgenic or genetically engineered animals. So mosquitoes carrying genes that prevent the transmission of viruses have been released in Florida. Now, let's walk through the process we would need to create transgenic animals. And we'll start by considering a mouse because this is a really common transgenic animal in science research. Now, when I say transgenic, let's break it down. Trans means across, and genic, we're referring to a gene. So in this case, we're talking about an animal that has a gene introduced from across an organismal boundary. It could be a gene from another mouse, but it could be a gene from a totally different creature, even a plant. So a transgenic animal is an animal that has been genetically engineered. Now, first in this illustration, we need to create the fusion gene construct. And in this case, we're looking at a gene that has a promoter and then a protein coding region. And this could be a promoter from one animal and a gene from a second animal. In any way, we'll be creating this fusion gene using restriction enzymes, DNA ligase, and usually bacterial transformation to make many copies of the gene. Additionally, we need a super ovulated female or a female who has been treated with hormones to produce many ovules at one time, many eggs released at a single time. Then we'll introduce this fusion gene into our egg cells, and those egg cells can be implanted into the mother mouse so that she can ultimately carry these transgenic babies to term. When those babies hatch or are born, we can perform DNA analysis such as PCR to look for the presence of the gene that we're interested in. And then only those transgenic offspring carrying our gene of interest will be bred further to create lines that are homozygous for our insertion. How is the introduced DNA integrated into the animal chromosomes? Well, first let's look at this cartoon. On the top here, we have the mouse chromosome, and you can see 4 exons are numbered, 1, 2, 3, and 4. The transgene vector that we're inserting, that is our DNA of interest, has a very similar sequence to the mouse chromosome. You can see that regions 1, 2, 3, and 4 are present, but we're adding in a new region of DNA, and in this case, it's neomycin, or a gene for neomycin resistance. That is a selectable marker, an antibiotic resistance marker introduced into the mouse DNA. Now, when the transgene and the mouse chromosome come together, because these regions are homologous or very similar, we can get homologous recombination or crossing over, recombining of the DNA, such that the mouse chromosome will ultimately be carrying our transgene of interest. And when this homologous recombination occurs in egg cells or in stem cells, the mice that are ultimately produced from this will be transgenic offspring. This type of homologous recombination, or flipping a gene into a specific location in the mouse chromosome, actually occurs very rarely, and we can also get introduction of this gene, our neomycin region, somewhere else in the genome of the animals. But homologous recombination allows us to target the insertion of our gene of interest to a specific known region of the animal's genome. To introduce our DNA of interest into a fertilized egg or an unfertilized embryo, we'll use a suction pipette to hold the egg in place while a tiny needle is injected from the other side, and this needle is containing our DNA, which can then be inserted in or near the nucleus of the egg. So we can take a male and female mouse, their gametes, egg and sperm, and combine the egg and sperm together for fertilization. And then before any further development has occurred, this fertilized egg can be microinjected with our gene of interest. That fertilized egg, or more likely that pool of fertilized eggs, is implanted into a mouse. And this mouse we're calling pseudo-pregnant because she has been convinced hormonally that she's carrying a pregnancy. The mouse carries these new transgenic babies and ultimately will be able to look at the offspring using PCR analysis to determine which one of them are carrying our gene of interest and which are not. How could you determine which of the individual offspring were carrying the gene of interest? Well, the answer is PCR. Since we know the sequence of the transgene, we can design primers to amplify only our transgene and not the genomic DNA of a regular mouse. When we run the PCR, it will amplify DNA only from those babies who have our transgene. And then ultimately we'll be able to run a gel and look for bands in the offspring. And in those offspring where we don't see bands, we know not transgenic. Other than microinjection of fertilized eggs, we could also transform stem cells. And in this case, we're using pluripotent stem cells that could become almost any type of cell in the developing embryo. So we have some embryo-derived stem cells that are growing in a dish here. We're growing them in tissue culture. And then these cells are injected or electroporated, treated with an electric current to introduce our small pieces of DNA. I'm drawing them as plasmids here, but they might also be linear pieces of DNA. Now, the cell that picks up our DNA of interest is not only going to have the gene that we're interested in, it's also got to have a selectable marker, so that we can screen for or enrich that one cell, that rare event of picking up our transgene. When we use our selectable marker, we can ultimately end up with a population of only transgenic embryonic stem cells. And these embryonic stem cells can be injected into a blastocyst. So we're looking here at a blastocyst, that is a developing embryo, and we're adding in our transgenic cells in the hopes that the offspring of this blastocyst will ultimately carry some cells with our transgene of interest in it. As with microinjection of fertilized eggs, these transgenic blastocysts can then be implanted into the mother, and the offspring will be chimeras. In this illustration, the transgene gives a black color, and you can see the mom is white, and this baby is almost entirely black in color, while the sibling is patchy. That is, there are regions of the animal that are carrying the transgene, and they're dark in color. And then there are regions of the animal that are not carrying the transgene, and they are light in color. This animal is a chimera. Some, but not all, of a chimeric animal's cells will contain the transgene. I would select this animal that seems to be highly expressing our transgene to breed for the next generation, and ultimately we'd be able to find animals that are entirely transgenic, no chimeras, and all of their offspring will also be transgenic. Let's talk about how to get homozygous mutant mice from a chimeric first individual. So all of our mouse pups that were produced from our transgenic event are going to be chimeric or mosaic. That is, some of their body will be black and some of it will be white, and different animals will have different patterns. Now, I don't really want to cross together two of these mosaic animals because they have potentially different DNA insertions. That is, the DNA may be inserted into different regions of their chromosome. So I'd rather start with one mosaic mouse or one chimeric mouse and cross it to a homozygous wild type mouse. Then some of the offspring will be heterozygous for our mutation that we've introduced. And we can continue to breed until we produce more than one heterozygote. And in this case, I'm showing them with dots. And from that heterozygous cross, we will get some individuals who are homozygous for our mutation. No longer chimeric, all the cells in their body will be entirely transgenic. This image shows you just how variant the expression of a chimeric gene can be. So these are mouse pups that are all from a pluripotent stem cell transformation. And you can see there's a lot of variability in how many cells in the mouse are expressing our transgene. We could say that these mice maybe have no cells that are expressing our transgene. Well, this guy, maybe he has 10% of his cells expressing the transgene. Maybe this one has 40% of the cells, 50% of the cells, 80% of the cells expressing that transgene. And it's only through selective breeding that we'll be able to produce animals where 100% of their cells are all expressing that transgene of interest. For just a moment, let's compare and contrast transformation of a fertilized egg with pluripotent stem cell transformation. With a fertilized egg, you know that the offspring is going to be entirely transgenic. That is, they won't have chimeras. However, because recombination is rare, that means that the offspring who is entirely transgenic may occur less than 10% of the time. So in a litter of 10 pups, maybe only one would actually be recombinant. Now, with a pluripotent stem cell transformation, we're going to have entirely chimeric offspring. That means virtually none of the offspring will have all transgenic cells. However, we're asking for this recombination event to occur in culture. And then we can select cells in whom that recombination event has occurred. We'll then inject these cells or introduce these cells into blastocysts, developing embryos, so that every offspring is going to be carrying our transgene in some proportion or another. You do have to go through generations of breeding in order to produce a homozygote. However, you have a lot more starting material. So there are trade-offs between using a fertilized egg when you get an entirely transgenic offspring to start, or using pluripotent stem cell transformation, where you get chimeric offspring to start, but you know that virtually all the offspring will be carrying the transgene. When we're talking about pluripotent stem cell transformation, how could you determine which embryos, which stem cells, had experienced recombination with the inserted DNA? It might be hard to use PCR. And that's because we're dealing with one cell events here. Single cell events, you can't just take DNA from the one, that's all the DNA that cell has. We want the one copy of DNA to use for PCR, but now the cell has no DNA. It's hard to do PCR that way. Instead, we might try and use a selectable marker that's in our DNA of interest, similar to what we would use in creating transformed E. coli cells. Can you come up with other strategies that we could use to work with these homologously recombined embryonic stem cells? I'm confident there are new ways of screening for these transgenic cells that scientists haven't invented yet.